Hello, my name is Alex Scott. Here is date. Let me get to my script. Okay, so I have chosen for my two cases, case number one and case number two. Case number one, part one. Have allosteric regulation plays a crucial role in controlling pyruvate kinase activity. One of the primary activators of pyruvate kinase is fructose 1,6-bisphosphonate, or F-1 6-BP, an intermediate of glycolysis. Fructose 1,6-bisphosphonate enhances the enzyme's activity by promoting its active conformation. Under normal glycolytic conditions, pyruvate kinase remains active, leading to the production of pyruvate. This pyruvate enters the tricarboxylic acid, or the TCA cycle, and subsequently the electron transport chain, or ETC, to generate ATP for cellular respiration. As with most enzymatic processes, pyruvate kinase is also regulated by inhibitors. ATP acts as an upstream inhibitor. When ATP levels are high, it signals to downregulate glycolysis, ultimately inhibiting pyruvate kinase to prevent excessive APT or ATP production. Another key inhibitor is acetyl-CoA, a product of fatty acid oxidation. In a fed state, the accumulation of acetyl-CoA suppresses pyruvate kinase activity to curb excess ATP generation. Additionally, alanine, the end product of amino acid metabolism, serves as an inhibitor. Elevated alanine levels also signal a slowdown in glycolysis to maintain metabolic balance. Phosphorylation is another regulatory mechanism of pyruvate kinase, particularly in the liver. Protein kinase A, or PKA, phosphorylates pyruvate kinase in response to glucagon signaling, which, which is triggered by low blood glucose levels. Phosphorylation inactivates pyruvate kinase, slowing glycolysis and promoting gluconeogenesis, a critical process during fasting in low glucose conditions, or in essence, not having anything to eat. Isoenzyme specific regulation further fine tunes pyruvate kinase activity in muscle and brain tissues. The M1 isoform rapidly the M1 isoform facilitates rapid ATP production, especially during exercise and other high energy demands. This is the normal process that the body goes through, is the M1 pathway. In the liver, the L isoform is regulated by hormonal signals such as insulin, which is an activator, or glucagon, an inhibitor. These balance the switch between glycolysis and gluconeogenesis. The M2 isoform is primarily involved in gene expression inactive in stem cells. Each isoform adapts to the specific metabolic needs of the tissue, ensuring efficient energy production and resource management. Overall, in total, the regulation of pyruvate kinase through allosteric effectors, phosphorylation, and isoenzyme specificity is essential for maintaining metabolic homeostasis across different physiological states. Many types of cancer cells are predominantly glycolytic and overexpressed on the oncoprotein MYC. Interestingly, the classical oncoprotein MYC has also been found to promote preferential expression of pyruvate kinase type M2 over the protein pyruvate kinase M1 activity. The isoform of M2 is characteristically found in low activity states and is ineffective at promoting glycolysis. That's when comparing the kinase type L or M1. Part 2 of the first case is understanding why cancer cells uh, prefer to go through the M2 pathway over the M1 pathway. Um, the main reason they do that is due to the M1 pathway not wanting to create the most biosynthetic way or the easiest way to make the body function. It is more focused on growth, development, and dispersion as fast as possible. So, getting into that part of the question, to understand the differential expression of M1 and M2 isoforms is essential to examine their distinct metabolic pathways. The M1 isoform promotes the conversion of phosphoenol pyruvate or PEP to pyruvate. This is a crucial step in glycolysis that efficiently produces ATP. That is the normal pathway that the body normally likes to go through to promote the most amount of ATP to feed to the cells to give them energy to grow. This isoform, referring to M1, is predominantly expressed in differentiated cells with high energy demands, such as those in the brain and muscles. The M1 isoform is less flexible, remaining largely in its active form, ensuring a steady flow of pyruvate and ATP production. This can be kind of broken down by, your brain is always working, so it needs the most amount of ATP to constantly be active so we can be functioning in day-to-day lives. And the same thing as muscles. We're constantly contracting and moving muscles doesn't necessarily have to be weightlifting or running or extraneous activity. Literally, what I'm doing right here with my hands is contractions of the skeletal muscles, and that requires a lot of ATP. So, looking at M2, in contrast to M1, this isoform exhibits reduced enzymatic activity and can transition between active and inactive states. This decreased activity slows the final step of glycolysis, causing an accumulation of upstream intermediates. These intermediates are diverted into alternative biosynthetic pathways, supporting cellular growth and proliferation. The metabolic shift underlies the Warburg effect, whereas in cancer cells, they rely on glycolysis for energy production, even in the presence of oxygen, prioritizing biomass generation over efficient ATP production. Is basically saying they, again, like I said before, prioritizing growth and numbers over actual proper biosynthetic pathways. So the reduced conversion of pyruvate in M2 expressing cells leads to an increase in lactate production, which is also mainly seen in skeletal muscles. This contributing to the acidification of the tumor's microenvironment. The tumor is just the overaccumulation of all the cell proliferation happening. And this acidic environment helps cancer cells to evade immune detection and promote their survival, meaning they can stay under the radar, continue to grow without phagocytosis of the cells to die to not grow more. The dimeric form of the M2 can translocate to the nucleus where it acts as a transcriptional co-activator. It activates hypoxia inductible factor 1 alpha, or HIF-1 alpha, which enhances glycolysis and regulates gene expression to promote cell proliferation and inhibit apoptosis. This inhibition of apoptosis and promotion of cell proliferation within the nucleus helps it to stay out of the detection from the immune cells, giving it a better or giving it a more easier way to grow and not undergo apoptosis. After it is created through the M2 pathway, post-transitional modifications such as the phosphorylation and acetylation of the M2 isoform enhance its tumor generic properties. These modifications support cancer cell survival by altering the microenvironment or the small, tiny environment that it's located in and facilitating metastasis. By keeping pyruvate in the cytoplasm, normally the pyruvate through the M1 pathway would go out of the cytoplasm and eventually make its way towards the mitochondria, and that mitochondria would eventually put it towards the electron transport chain to support the promotion of ATP for other cells. It enhances the tumorigenic properties of keeping it within the cytoplasm. With these modifications to keep it in the cytoplasm, this supports cancer cell survival by altering the microenvironment again. With keeping the pyruvate in the cytoplasm, then rather anchored in mitochondria, the M2 expression favors cellular growth and longevity over its efficient energy production as it would be in the M1 pathway. Overall, the distinct regulatory mechanisms of the M1 and M2 isoforms highlight their specialized roles. M1 supports efficiency and energy generation, while M2 drives metabolic adaptation in proliferative cells, particularly cancerous tissues. So that is my first case. The second case, I chose case 2 with the 16-year-old female. So with the first question, she has a deficiency of muscle glycogen phosphorylase, and I am essentially the physician, and they're asking me the questions. The first question is stated towards me that she is saying, I am not sure I understand the issue. Can you explain how muscle glycogen is used normally during exercise? Skeletal muscle stores glycogen, a form of glucose, which serves as the primary energy source for muscle activity, just any types of muscle activity of contraction. During exercise, glycogen is broken down through a process called gluconeogenesis. This breakdown releases glucose, which is then used to produce ATP, the energy molecule that powers the contractions and bodily movements. So that is basically me explaining how glycogen is normally used during exercise, so that glycogen is broken down to glucose, which that glucose is then the able-bodied form of energy production to downstream promote ATP production for those contractions of the muscles. And then question 2, to simplify the concept, think of your body as a gas-powered car. The body of the car represents your body, while the wheels symbolize your muscles. Just as a car needs fuel to move, your body relies on glucose for energy. When you start the car, gasoline travels through a complex engine system similar to how glucose moves through gluconeolysis. Pressing the gas pedal activates the engine, converting fuel into energy to turn the wheels. In the same way, glucose is metabolized to produce ATP, which fuels the muscle contractions. Part C, when glucogenolysis slows down, less glucose is available. This reduces ATP production. This then limits the energy supplies for the muscle movement. Just like a car running low on fuel, efficient regulation of glycogenolysis is crucial for maintaining energy levels and supporting physical activity. When referring to her long walks with her friends, she will be able to go and enjoy long walks with her friends. As I had stated above, the muscle contractions and energy production for your muscle glycogen phosphorylase deficiency limits your body's ability to fully access the glycogen stores for ATP production. However, your body will adapt by shifting its primary energy source to fat oxidation. The fat stored in and around your muscles is known as brown fat. This will become activated and begin releasing its energy to fuel your activity. This allows your body to continue functioning, relying on this alternative energy pathway to support your movement. So the brown fat or the fat around the muscles or throughout the body are mainly used to store energy, just as glycogen is stored uh, in a normal functioning human without this glycogen phosphorylase deficiency. Again, like I had said before, the glycogen normally will be synthesized into glucose, which is the active form, and that glycogen is the storage form. Glycogen is just like the brown fat where it has the stores, but since she has this deficiency in the glycogen phosphorylase, glycogen cannot be phosphorylated to glucose. So the brown fat will instead be phosphorylated to downstream use ATP for its movement and energy production. When it comes to eating many meals, or especially eating at night to maintain blood glucose levels, the muscle glycogen phosphorylase deficiency specifically affects your skeletal muscles, which is where that glycogen is stored. It has no effect on your liver within the blood glucose levels. Since the liver is mainly supports blood glucose, as we've seen in different types of diabetes, um, glycogen is mainly stored within the muscles. So not eating at night won't have any effect because your blood sugar will remain stable even while you are asleep, a state known as fasting, which we had learned in previous chapters. Within the liver, the enzyme glucokinase plays a key role by isomerizing glucose into glucose 6 phosphate, which that part is essential for maintaining proper blood glucose levels during periods of fasting. That biochemical pathway is not correlated to the glucagon or glycogen glucose phosphorylation through skeletal muscles. When it comes to lactate production, as we know, over exertion of muscles leads to lactate buildup, and that's what causes that soreness in our muscles. And during anaerobic activity, which is the absence of oxygen, glycogen is essential for producing glucose, which is then used to generate energy for the muscle contractions. In normal functioning cells, glycogen is converted into glucose, which undergoes glycolysis to form pyruvate. This pyruvate enters the TCA cycle, leading to ATP production for energy. The byproduct of this ATP production is lactate through anaerobic glycolysis. However, since her body is unable to perform anaerobic glycolysis due to muscle glycogen phosphorylase deficiency, it won't be able to produce any or little lactate, as we would typically see during intense exercises. However, since we're not producing as much lactate for the output of pyruvate that we're producing, she will have a faster muscle fatigue than the normal person would, as the one discussion we had of the swimmers. The second swimmer who was able to walk around the island but was not able to swim out to the boat as fast. That is because he's not able to produce the lactate. So there's no byproduct. So it's just a buildup, and that causes the not the breakdown, but the inhibition of the muscles to contract like they normally would. That is my spiel. Good night.